All right, so one preamble that I didn't provide before the replication lecture is this entire unit is focused on the central dogma, the idea of from DNA we get mRNA, and from mRNA we get protein, and that so for last lecture there's also the notion that from DNA you can get more DNA during replication. So today's lecture is going to be discussing the entire process of, or one half today's lecture. We're going to have transcription today and on Thursday. The first half that we're going to go over today is setting the foundation for transcription, how we go from DNA to mRNA. And we'll be discussing this in the context of diabetes. And then when we get to transcriptional regulation next class, we'll be applying that in the context of the use of anabolic steroids. So diabetes mellitus, or as we just commonly refer to as diabetes, bless you. It's a categorization of a group of diseases that are characterized by hyperglycemia. And so the way in which this manifests can kind of differ between the different types of diabetes, but it can be characterized by either insulin defects in insulin secretion, insulin resistance, or both. And the mellitus in the clinical term, diabetes mellitus. Mellitus just is a Latin word for sweetness, and we will discuss why. And it's not for the reason why you think when it relates to hyperglycemia. So the cells, all of our cells have insulin receptors on them. There's some cells in our bodies that have slightly more, but all of our cells, because of the bioenergetics required to carry out their functions, have receptors for insulin on them. And so when we consume or take in glucose, there's an insulin, there's a release of insulin, and our cells in our body have insulin receptors on them. And so what happens is that when insulin binds to the receptors in our cells, they increase expression of glucose transporters so that glucose can be shuttled into our cells. And so once glucose is shuttled in, then it can be used as a substrate through a few metabolic processes to produce the key energetic substrate, ATP, that's key for a lot of our fundamental processes in our cells. And so the chronic hyperglycemia of diabetes, the hyperglycemia means elevated sugar concentration in our bloodstream. This is associated with a lot of long-term pathologies, and some of it is involved with the dysfunction of some key organs and tissues, especially the eyes. There's diabetic retinopathy, basically the deterioration of the retina as a result of diabetes because of the poor circulation. So the smallest blood vessels in our bodies are called capillaries. And, sometimes if you look in the mirror, you can actually see some of these like small capillaries in your eyes. This elevated blood sugar starts to affect the capillaries and the ability for proper blood flow in these small capillaries. And if there's not proper blood flow in your retina, it'll lead eventually to diabetes-induced blindness. This can also affect the kidneys, diabetic nephritis. There's also issue with nerves. Diabetic neuropathy. There's actually, again, like all of our cells need nutrients that's provided through blood flow, and these small capillaries are affected that affect the abilities of these nerves to work properly. And so typically patients that have really severe cases of diabetes that's left unchecked and untreated, typically, we'll talk about the different treatment pathways here, but without proper treatment, there's diabetic neuropathy, so they can't feel things properly, which often leads to either really severe diabetic ulcers or bed ulcers if they're not mobile sufficiently. And eventually, this will lead to really severe heart issues, as well as, again, really severe pathologies with the blood vessels. So some of the symptoms associated with diabetes the first couple are polyuria and polydipsia. As you can imagine, this hyperglycemia, this high this high blood sugar concentration in our blood, when it's filtered out into our urine, it creates this osmotic pressure that's driving fluid loss through urine. And so there's typically, when people are first being diagnosed, you'll see when people are first being diagnosed with diabetes, there's some of the first symptoms that they'll look for is polyuria. Polydipsia is another one, this constant thirst. And so one thing I forgot to say that I think is associated with the mellitus part, doctors from long ago, one of the ways in which they would diagnose diabetes, again, there's high sugar content that's in the blood, it's hyperglycemic, sugar's being lost into the urine. One way that they would diagnose diabetes is they would taste the urine, and if it was sweet, this person's diabetic. Thankfully, we have better diagnostic tools now, but long ago, that was one diagnostic tool for diabetes. Polydipsia, that there's this loss of water because of this osmotic gradient on the urine, so they're constantly also thirsty as well. There is a general nutrient deprivation as described by the weight loss and impaired growth. And there's also additionally ketoacidosis. And so that what that ends up happening is that there's the breakdown once if there's not sufficient availability of sugar, of glucose being taken up into cells, they'll look for other substrates as a source of energy. And so they'll start to break down lipids. And so once lipids start to be broken down, they'll create what are called these ketone bodies. And so these ketone bodies, once enough ketone body concentration gets high enough, these patients will enter into ketoacidosis. You know, their breath will actually start to smell kind of fruity as well. And so that's typically another symptom of patients who are diabetic. We've already talked about a few of these, but the long-term complications of diabetes, there's the retinopathy, the capillaries in the retina leading to the capillaries being affected, poor blood flow that leads to deterioration of the retina and blindness. There's diabetic neuropathies. Neuropathies is poor nerve function that leads to some serious ulcers because they literally can't feel some issues, whether typically in their extremities. So the capillaries, these small capillaries, are everywhere in our bodies, but oftentimes, as you can envision, are usually in our digits and extremities. And so that's usually where these ulcers will present, particularly in the feet. There's going to be, bless you, increased incidence of cardio and cerebrovascular diseases because, again, these rely on, particularly cerebrovascular diseases, rely on the capillaries. And if there's any dysfunction with them, that'll lead to some pretty severe pathologies. And as you can imagine, because of the effect on metabolic function as well as on vascular function, this will lead to hypertension, which is high blood pressure, abnormalities of lipoprotein metabolism, because now they're usually switching over to metabolizing lipids as a energetic source or substrate. And there's also oftentimes periodontal disease. So this is usually like some sort of disease with the gums. And so that's oftentimes attributed with because of this excess of sugar that's present in the bloodstream, and there's a lot of surface capillaries within the periodontal space. There's a lot of bacteria that thrive on sugar that will then like kind of like change the like microbiota of your gums, and oftentimes will lead to some sort of bacteria-derived periodontal disease. So as you can imagine, of the things we've discussed up to this point, this might be a clinical application that has some of the highest prevalence and economic impacts. And actually, I have some updated numbers that I did not change on the slide, so I'll be saying them here on the slide, be providing them or updating it here. So for the economic impact, so this was as of 2017. Oh, sorry, I did update it. Forgive me. So as of 2017, I couldn't find updated numbers for 2023, but the total cost, that means direct and indirect effect, is $327 billion in the United States alone. And the direct effect, which means like direct clinical expenses, is $237 billion. I suspect, based on how I saw numbers go up from 2012 to 2017, that these numbers have probably gone up on the order of, I'm going to ballpark like 450 billion in total costs approximately, and close to 320 billion for direct costs. That's an approximation, assuming proportional increases from 2012 to 2017. In the US alone, there are 1.9 million new cases of diabetes per year. And that is likely to continue, that likely will probably have changed in the last 5 years as well. There's also a bit of a demographic difference across the US, where it's typically rare in African and Asian nations. And there's a few different possibilities as to why that's the case. It also varies by race in the US, from 7 to 14% between particularly white, Black, and Hispanic communities. What's interesting is that there is a genetic predisposition in some cases of diabetes, where there are some communities like the Pima and Papago Native American tribes, where rates are at up to 50% of those have diabetes. And so we typically think of only the prevalence of diabetes as a result of poor diet and lack of exercise, but there's also a genetic, there's also the autoimmune component as well, but there's also a genetic predisposition where it's really present in some communities here in the United States. Before I jump into transcription, or sorry, I forgot, there's one more slide I was going to go into. So we have a few different types of diabetes that we're going to talk about today. The first is type 1, which is the autoimmune kind, where it was the key insulin producing cells in our bodies are these pancreatic beta cells. And so what ends up happening is that in type 1 diabetes, there's a loss of these type 1, there's a loss of these pancreatic beta cells, the insulin producing cells. And the way in which it happens is that there's some sort of antigen that's recognized as foreign on these pancreatic beta cells. And so there's an autoimmune response that promotes the destruction of these beta cells in these type 1 patients. And so it was historically called juvenile diabetes. I'm guessing probably most of you were probably not around back when it was called juvenile diabetes. I think that was like back when I was a kid, and it kind of switched because its emergence doesn't always happen just in juvenile. So typically diagnosis for type 1 diabetes can be in the range of like 8 to 10-year-olds, but there are also times in which it's diagnosed in patients who are in their teens, even late teens. My first year dorm mate here at UVA, he was diagnosed with type 1, and literally polydipsia, polyuria, severe weight loss. And so it can happen sometimes as late as your late teens. Why that's the case, I don't know. But typically, it's typically diagnosed in patients who are in the range of like 8 to 12. Another form of diabetes is type 2, which historically was just called mature onset diabetes. This is a much more prevalent category than type 1. And so this is the variation of diabetes that's typically associated with poor diet, lack of exercise, and obesity. And so one of the reasons why perhaps African and Asian communities do not have as high rates of diabetes is that they have very different diets as well as very different lifestyles. And so the type 2 diabetes typically is a result of improper regulation of insulin signaling, as typically attributed to insulin resistance. And so but what's interesting is that another version of type 2 that's typically less common but still key for our lecture today, and we're going to talk a little bit more on Thursday, is mature onset diabetes of the young. And that's typically a transcriptional defect resulting from some sort of inherited mutation that leads to dysfunctional insulin signaling. There's also another form of diabetes that's not typically talked about less, is sometimes dubbed type 3 gestational diabetes that can occur during pregnancy. And so that one is far less understood, but there's a belief that perhaps really dramatic hormonal changes during pregnancy can predispose one to dysfunctional insulin signaling that leads to diabetes. And there are cases where multiple bouts of gestational diabetes can actually lead to type 2 diabetes itself. With that, are there any questions before we jump into transcription? Yes.
Cells is the last of beta cells. So then type 2 is just that there's beta cells, but they are not properly producing insulin. Is that correct?
There is it's not that they're, yes, there's this alteration where there's this resistance to being able to produce the proper amount of insulin to meet the high demand of really high blood sugar levels. So like typically blood sugar levels should be, depending on if you're fasting or if you just eaten, but like about 100 is like a good level. And so sometimes if it's as high as like 400, like that's really hyperglycemic. And it's hard to meet that metabolic demand of producing enough insulin. And so that's why oftentimes people will try and balance their blood sugar levels, or if need be, get an insulin injection if they need it. And so there's this like dance of like not being able to produce enough insulin, but also being resistant to insulin because their blood sugar is so high all the time. Does that make sense?
Yeah, I see. So insulin, its whole function is to combat high glucose level. So if your blood sugar is constantly, then every injection you get of insulin, your body's going to build up a resistance response to that insulin.
Let me provide one point of clarity that could help. So with type 1 diabetes, it's an autoimmune disease where the pancreatic beta cells are being destroyed. So they don't have the cells that are producing insulin. Yeah. They respond to insulin properly when it's present, but it's just that they don't have the cells that make it. And so typically, patients with type 1 diabetes will get insulin shots. They'll constantly, there's also now, I can't remember what BME group made it, but there's like basically an artificial pancreas where they have a sensor that both detects their blood sugar level and injects insulin. And so like what they need is insulin. Typically with type 2 diabetes, it's this need to constantly monitor blood sugar levels and provide insulin if needed. But it's really this resistance to insulin where the sugar in the blood is not being shuttled into cells properly. So their blood sugar is constantly high. So they need to monitor diet and exercise to maintain proper blood sugar levels, because if it's too low, it can be super problematic. And if it's too high, it can be super problematic. So it's this matter of needing to monitor it as opposed to really providing insulin like with type 1. Yeah.
So then is type 2, it has nothing to do with beta cells. All beta cells are present, but then it's just the insulin, something is misfunctioning, malfunctioning with the insulin. It's secretion of insulin.
Not the secretion, but rather the response to insulin by the cells, because they're resistant to insulin for some reason. And so what they need to do is typically monitor blood sugar, regulate their blood sugar levels more closely. With type 1 patients, they need to monitor their blood sugar levels, but they can take insulin injections to make sure there's constantly enough insulin to take up for cells to take up the blood sugar that's available. All right. Yeah. So we're going to move to try and describe what are the stages or pieces of transcription. Excuse me. What are the stages of transcription? And we're going to look at it. We're going to try and provide a couple of distinctions between prokaryotic and eukaryotic transcription. And then we're going to try and talk a little bit about the pieces that are involved and and how these transcripts, the subsequent mRNA transcript that's made, and how it goes from like a immature to mature form. So some of the key pieces of transcription is we have a transcriptional unit, which is one gene. So in eukaryotes, I should say, one transcriptional unit as designated here from promoter to terminator is typically one gene. In prokaryotes, one transcriptional unit does not equal one gene. There's oftentimes multiple genes present within one transcriptional unit, but that just means those genes are all regulated together. And so they all kind of work together in some capacity. But the key thing here for the first point is that in eukaryotes and us, one transcriptional unit from promoter to terminator is one gene. For prokaryotes, it's not just one gene. The promoter is the site at which transcription begins, where we have our RNA polymerase here that binds at the promoter region. And so unlike DNA polymerase, so DNA polymerase needed a lot of help. DNA polymerase is able to elongate an existing DNA strand, but it couldn't open up it couldn't unwind the DNA. It needed help with supercoiling. But here with RNA polymerase, we don't have helicases, we don't have gyrases, we don't have supercoiling. The best example I could try and give here, like a mental example, I don't know if this is going to sink home with you guys, but the distinction between like RNA polymerase and DNA polymerase, if you've like lost something in the couch, like RNA polymerase is basically like your hand going in the couch and going through, whereas DNA polymerase is like pulling things out, like you're pulling out the cushions of the couch and going in. And so like RNA polymerase is like sliding into the double-stranded DNA, unwinding it a little bit and going through from promoter to terminator. So again, promoter is where transcription begins. The terminator is where transcription ends. And so what ends up happening is that using these monomers, these NTPs, these nucleotide triphosphates, ribonucleotide triphosphates here in this case for transcription, it's able to use those monomers to build a mRNA transcript using these monomers, and it grows the mRNA in the 5' to 3' direction, just like with replication. So what is being produced in the 5' to 3' direction. So that means it's reading this unwound DNA. It's reading, we have two strands here. The bottom one is the template strand, what it's reading. So it's reading the 3' end and creating a complementary 5' mRNA and creating it in the 5' to 3' direction. So typically we have the template strand and the coding strand. The template strand is being used as the template to create a complementary and anti-parallel mRNA. The coding strand, because it's also complementary and anti-parallel to that template strand, your mRNA that's being produced here and the coding strand are going to look exactly alike, except for the mRNA, instead of T's, you're going to have U's. You're going to have uracil in your mRNA. Nothing here is happening to the coding strand. It's just using the template strand. At the very end of the lecture, I'll talk about what's really unique about the coding strand that is kind of based on how transcription happens within eukaryotes. But for the context of where we're at in this lecture right now, nothing is happening here with the coding strand. The template strand is being used for transcription here. So we have initiation that happened at the promoter, at the initiation site. And so during elongation here, you're going to have a growing mRNA strand here that's growing from the 5' to 3' direction, using ribonucleotide triphosphate monomers to help feed in the growing strand of RNA that this RNA polymerase is going. And so it's going to continue elongating. Once it hits this terminator, there's going to be a termination signal that is communicating that this is the end of this transcriptional unit. This is the end of this gene. And there's going to be a couple things that happen that lead to the cleaving of the mRNA strand here. And once that leaves, that's actually going to induce a conformational change to RNA polymerase 2 to then leave, and it's going to leave the double-stranded DNA there, and you'll be left with this mRNA transcript that's been made in the 5' to 3' direction. And so the key thing to take away here is that in the terminator sequence, there's actually an encoded sequence that serves as a signal for the cleaving of the transcript and for the release and leaving of the RNA polymerase. So first, I'm going to take a second to talk about promoters, that start site where transcription begins. First, I'm going to talk about bacterial promoters. I'm going to talk about, after that, eukaryotic promoters. So with bacterial promoters, there's going to be a specific consensus sequence in the template strand. And so we have what we have here is the entire promoter here. And this is the promoter, again, is where RNA polymerase binds. And so there's these multiple consensus sequences at which that are upstream of the start point. So we have our start point here. That's the T on the template strand. Everything upstream of it is going to be characterized as the promoter, where RNA polymerase binds and is going to initiate transcription. And there's specific consensus sequences here within the promoter. And similar to the consensus sequences, so up to this point, we've only applied the phrase consensus sequence to the origin of replication during replication. Moving forward now, you don't need to only consider consensus sequence as a phrase applied to only origins of replication in prokaryotes, but rather think of it as these conserved sequences of nucleotides, that these repeating patterns that exist within our DNA that help serve as like a signal point for something to happen here. And so similar to with origins of replication, as like we discussed before, here for the promoters, there's bioinformatics work that was done to define what are the consensus sequences within bacteria, within prokaryotes, for what their promoters are and what are the common features that define these consensus sequences. So now moving towards eukaryotic promoters, what ends up happening is that at the initiation here, one thing I forgot to say, forgive me, is that typically for initiation, this is true both with bacterial promoters and with eukaryotic, and we're going to talk about the eukaryotic one in a second, is that there's this ta-ta box that's one of the consensus sequences within promoters. And so we're going to show a little bit later why that ta-ta box is so important, but that's a consensus sequence. So during the initiation of transcription, what ends up happening is that RNA polymerase will bind, and it'll actually stay put for a bit as the RNA DNA hybrid, as there's the initial formation of the mRNA transcript. And once that happens, then RNA polymerase will take off down the DNA strand, producing this mRNA transcript using these nucleoside or ribonucleoside triphosphates. These are the monomers. These are the building blocks for the new growing mRNA transcript. Just for clarity's sake, the way we refer to this growing mRNA, you can say growing mRNA strand or mRNA transcript, or you can just say transcript because it's what is being produced from transcription. And so one key thing to note here is that when the RNA polymerase is coming in for transcription, it doesn't need a helicase and it doesn't need gyrases, because when it comes here, there's no supercoiling that happens. And so there's no requirement to, there's no supercoiling, because no supercoiling is occurring, there's no gyrases needed. So during elongation, RNA polymerase is moving down double-stranded DNA and slowly unwinding it as it goes along. And as it moves down, it's because there's no helicases leaving open or single-stranded binding proteins. Once RNA polymerase moves down the DNA strand, what was unwound before comes back together. All right, and so it's basically sliding through. And this is why I tried to use that illustration of like your hand going through a couch, where it's like you don't need to like pull things out. You're sliding through. That's kind of like what RNA polymerase is doing here. And it's adding ribonucleoside triphosphates to the growing mRNA strand in an anti-parallel but complementary fashion to the template strand. Again, it's using the template strand as its template. And it's going to be what's produced is going to be matching to the coding strand. And so if you, bless you, if you looked up on a bioinformatics website for a specific gene of interest, you could look at the coding strand of DNA, and you could look at the known mRNA transcript for that that's produced from that gene, and they would match up. But the only difference would be instead of T's, you'd have U's there. And so the DNA rewinds back together behind RNA polymerase as it moves. And again, it is moving in the 5' to 3', or sorry, it is growing the mRNA transcript in the 5' to 3' direction along the template strand, which is in 3' to 5'. So the 5' to 3', it's pretty similar to replication, where the new DNA strand that's being produced during replication is being produced in the 5' to 3', similar with replication here with RNA polymerase. And finally, we have termination at the terminator site. What ends up happening is that there are specific sequences encoded within the terminator that actually do something pretty cool here. What they end up doing is that they actually encode this self-complementary sequences, whereby what ends up happening is that the resulting mRNA transcript will begin to form a hairpin loop because they're complementary. So that means that perhaps there was a CCCCCCC region, some region of transcripts here, or some nucleotides here, and then GGGGGGG. And then these two sequences are complementary, so they'll form this hairpin structure. This hairpin secondary structure actually provides like a mechanical cue to RNA polymerase that this is the terminator region and transcription needs to be stopping. And so it's the hairpin structure is a key feature of mRNA that serves as a physical and biophysical cue for the end of transcription. I'm going to stop there for a couple of questions. Yes. Correct. It's encoded in the terminator sequence of the DNA, but it is what ends up happening is the hairpin is the mRNA itself, where it's complementary and loops back on itself because of what's encoded in the terminator in the DNA. Yes.
Okay. Several slides ago, I think it's the second stage of the transcription slide, you said there was something important to take away about termination. I just didn't catch it.
Yeah, so what I was saying was that there's it actually is right here. That's actually, it was referencing vaguely what we talked about in this slide. So what I said a few slides ago is that the terminator sequence, so the promoter is like the initiation site of transcription. The terminator is where transcription stops. The terminator has an encoded signal that helps signal for RNA polymerase to stop transcription and to unbind from the DNA. That signal in the terminator sequence is actually this terminator sequence that produces this hairpin structure. So this hairpin structure, it's actually like it's like this physical structure that serves as a cue for transcription to stop. Yeah.
On slide 8, you say that promoter is upstream on the coding string.
I think I know what I meant to say. If that's what I said. What I meant to say is that the promoter. I'll actually go back to that slide. What I was saying is that the promoter region. So we have this entire green area is the promoter, but right here it says start point. This is where transcription begins. But upstream from that, back before that, on the template strand, is the promoter region. So we have our start point where transcription will go down that way, or RNA polymerase will move down the coding strand that way to the right. But just before the start point, if you move to the left a little bit, that's the promoter region that will actually be the site at which we're going to talk about it in a couple slides, but the site at which a complex of proteins will form to recruit RNA polymerase to begin transcription at the start point.
I'm sorry, I get that clarification from the start point, but I don't think I fully understood the question. Could you clarify that, or like, I didn't understand like their question. Or kind of what the question was.
I think his question was born out of something I may have misspoken when I was trying to say that, excuse me, to the right, it says start point over there to the right. And I was trying to describe how upstream from that, to the left of that on the template strand, is a region of that gene called the promoter, where a complex of proteins will form that'll recruit RNA polymerase, and then transcription begins at the start point, but it's initiated, it is prompted by the promoter to the left of it, just upstream, for these other proteins to form. And we're going to talk, the reason I'm saying like these protein complexes, we're going to talk about that in a second. But the key thing to take away that I was trying to describe here is that the promoter is actually just before the start point. That's the key thing to take there.
And then the start point is for initiated.
The start point is just like the first nucleotides where transcription begins. I'm going to go over here first, and I'll last two questions here.
How many like base pairs is the terminators?
That varies between prokaryotic and eukaryotic DNA. Shoot. I don't know off the top of my head. I know it varies, but I'll get back to you on that. I'll go here first.
So for elongation, it says that it's adding about 12 base pairs of RNA haplotypes, but then it adds 12 through at 12.
Yes,
Not
correct. Yes. Last question.
Just to clarify, so the RNA polymerase moves from three, or like leads to the side prime, but the
Yes.
strand being created is going by.
Yes, because it's complementary and anti-parallel. Perfect. All right. Okay, so with eukaryotic transcription, we have 3 different kinds of RNA polymerases. And as you guys have already discussed, I think before this class, while I was gone, there are 3 kinds of RNA. And so they kind of, what's nice is that there's a RNA polymerase species for each type of RNA. The key one that we're going to talk about today is RNA polymerase 2, which is what makes mRNA, or particularly the pre-mRNA, that will then be matured into full mRNA. But it's good to know that RNA pol 1 makes ribosomal RNA, and RNA pol 3 makes tRNA. But the one we're going to focus on, the key thing, and it's in blue, RNA polymerase 2 is what makes mRNA. And that's the key one. That's what's key for transcription. And each polymerase also recognizes a different promoter structure. So for RNA polymerase 2, the promoter that it identifies, there's going to be a few key features, but there's going to be one thing that I want to draw your attention to. So there's going to be this entire initiator sequence around the starting point. So the starting point is this adenosine right here that's labeled +1. And there's going to be pyrimidines to the left, and there's going to be 5 pyrimidines to the right that serve as like the initiator sequence around that start point. But the key thing to note here, and so forgive me, these pyrimidines can be T's or C's, and so it just means there's going to be 2 of them, 2 T's or 2 C's, or some combination with a C, and there's going to be 5 of them to the right. But the key thing that I want to draw your attention to is that there's going to be a TATA box to the left of that initiator sequence. And so this is going to be about 25 base pairs long, but that TATA, the TATA, is a key feature of the promoter. So the TATA box plus the initiator, these sequences around that start point, that constitutes what we call the core promoter. There's so much variety outside of that within a promoter. So if we have the BRE, which is the B recognition elements, we have the DPE, which are the downstream promoter elements. Those have so much more variety that it's hard to know what they are from gene to gene. But what we do know, what is like a consensus sequence, for lack of a better phrase here, is that within the eukaryotic promoter, within the eukaryotic core promoter, we have the TATA box, with these initiator sequences, with these pyrimidines around the start point. And so what's key to note here is that there's a class of proteins that bind to DNA that help initiate transcription called transcription factors. You guys may have already heard about them during your research or come across them. What these proteins do is that they'll be in the cytoplasm, they'll get phosphorylated by kinases, they'll move into the nucleus, and they'll bind to specific genes. They'll bind at the Tata box before RNA polymerase II. They serve as a way to increase the likelihood of RNA polymerase II to bind to promote transcription. So the more transcription factor activity you get, more likely you're going to get transcription of that gene. So we have our core promoter that has the Tata box and initiator sequence, and transcription factors, these proteins that bind to DNA, bind to the Tata box even before RNA polymerase II does. And they help recruit RNA polymerase II. What's really key here is that there are some transcription factors that don't need to be activated by some kinase. We have these, what we call basal transcription factors, that is always required to allow RNA polymerase II to bind to DNA. And so for RNA polymerase II, that basal transcription factor is called TF2D. Excuse me. And so what that means is that if that promoter is open, if it's available, TF2D will always bind. That's one key thing I want you to know, is that this is a basal transcription factor. And what that means is that if that Tata box is available, one thing we haven't quite talked about yet that is becoming kind of more of a standard thing in cell biology is that our DNA, it's these chromosomes are kind of compressed and compacted. If our DNA in a certain gene location is compressed and compacted, that it's hard to access that region of DNA to actually promote transcription. But if it's open, if the chromosome is open in that region, that increases the likelihood of transcription. And so if that gene is open, if that Tata box is available, the basal transcription factor, TF2D in this case, will always bind. And so the more that what ends up happening is that once that binds, more transcription factors can actually come in and bind. There's so many different kinds of transcription factors. Different scientists will have their favorite one, their pet transcription factor that they study. But through protein-protein interactions, more transcription factors can come in and bind. And the more that bind, the more that increases the likelihood of RNA polymerase II to come in and bind and promote transcription. And so, again, the key thing to note here is that basal transcription factors are a key necessary part of transcription. And if the Tata box is available and open, they will bind. And it will bind by first finding the Tata box. And it can recruit other transcription factors, but you need TF2D first to then recruit RNA polymerase II.
No.
Perfect. I read your mind. And then you'll get transcription of RNA polymerase II moving down the gene to promote transcription. So once transcription occurs, we have our mRNA transcript. There's a few key things needed to modify it from its pre-mRNA, this like immature form, to a mature mRNA transcript. The reason why that's important is because for any of you that may have already done, whether it was in Ideas Lab or in labs you're working in, you may have already discovered that mRNA is really unstable and easy to degrade. If any of you have had the chance to do a qPCR, they can be really finicky. And so there are things done to mRNA to help promote their stability so that they're not easily degraded. And we're going to talk about what they are. So there's a 5' cap, there's a poly A tail, and the last thing we're going to discuss is splicing events that you may have already talked a little bit before on the lecture that I missed. But we're first going to talk about the 5' cap. What ends up happening is that, so again, just to remind you where we're at, we have our pre-mRNA. We have an mRNA strand that has been produced from transcription, and it's going to be modified now. So there's this cap that's added on, this backwards 5' cap. That's this methylated guanine. It's a 7-methyl guanosine. 7 refers to the location for some of these modifications. And what ends up happening, it's backwards because we usually move in a 5' to 3' direction. It's added in a 5' to 5' direction. And what's really key is that that backwards nature of it is really key for some other proteins that are coming in and bind. But the key functions for this 5' methylated cap is that it's a signal for a nuclear export. So when our mRNA is matured, we want it to move out of the nucleus so that it can be used to make proteins. It's also going to be helped. It's going to be a huge major help with for being a site at which certain proteins will bind at the cap to help with the stability so that it's not degraded. And it's also a key binding site for ribosomes for when it's outside the nucleus for when we're going to make proteins with it during translation. But there's a key capping enzyme that mediates this 5' methylated cap to be added that only associates with products from RNA polymerase II. Why do you think it's important that this capping enzyme is only associated with what's made from RNA polymerase II? It's not a trick question, but just like as like an application, like why do you think that's important?
Does RNA polymerase II only interact with eukaryotes, so only
You're on the right path, but it's not just that. It's not about only eukaryotic cells. Think about what's unique to RNA polymerase II as compared to the other RNA polymerases. Yes, exactly right. Because we only want mRNA to be a binding site for ribosomes, a binding site for nuclear export. So like it's a key, it helps serve as a key feature for mRNA so that it can be used for translation when it's used to make proteins. In addition to having 5' cap, mRNA is modified via a process called polyadenylation. That can be a bit of a mouthful, so you can say a poly-A tail. So what ends up happening is that our DNA, our genes vary in size. And what ends up happening is that genes can be a few kilobase pairs long or 40 kilobase pairs long, 40,000 base pairs. And transcripts, because the terminator is some sort of like physical cue because of the hairpin loop, sometimes it can take a bit for termination to finish. And what ends up happening is that we just get a lot of extra unnecessary, our transcripts become too long, for lack of a better phrase. I'll stop stumbling around. I'll say our transcripts are generally too long. So there's a need for clipping that, but also a way to mark the end of like the important information within our mRNA transcripts. So what ends up happening is that there's a specific signal. Now it's about 10. That's another consensus sequence here. That this is a AAU. That's a poly-A signal. It's a signal to this enzyme called poly-A polymerase. You can tell by its name that it's a polymerase that adds poly-A tail or adds polyadenylates. And so what ends up happening is it is a mark for like the end of the important information in this mRNA transcript. So what ends up happening is that poly-A polymerase or PAP does a couple of key functions here. Once it sees that poly-A signal, this AAAUAA, what ends up happening is it cleaves the transcript just a little bit downstream from that signal. And what ends up happening is that in addition to that, it adds a poly-A tail. It adds many adenosine to the tail of that mRNA transcript. The key reason why that's important is that the poly-A tail helps protect the transcript from degradation. There are these like poly-A binding proteins that help stabilize the mRNA. And it's also a key necessary part for the full initiation of translation once this is like shuttled out and used to make a protein. The length of the poly-A tail can vary between prokaryotes to eukaryotes. For prokaryotes, I think it's about like approximately like 20 to 30 nucleotides long, this poly-A tail. And eukaryotes, it differs from gene to gene. It's about 20 to about 100 nucleotides long. But the reason, another thing to consider is that a possible evolutionary explanation for why it's A and not C, C, U, or G is that the monomer in ATP adenosine is like it's this nucleotide, and so like there's potentially like this wide availability because that's the key substrate for energetics that it can be used to create the poly A tail. That's just one explanation as to why A for the poly A tail. But yeah, so what ends up happening here is that we have our 5' cap designated by G here, this 7-methylguanine. We have our poly A tail with these poly A binding proteins. And what ends up happening is that they come together to ensure that the ends of the mRNA are not available for exoribonucleases. So again, an exonuclease will degrade, will take away nucleotides at the ends. So, and there's a large number of exoribonucleases within our cells because viruses are single-stranded RNA that our exoribonucleases degrade. And so like it's a defense mechanism, but we also need to make sure that it's not targeting our mRNA that we want. And so the poly A binding proteins help ensure that that this mRNA transcript is protected. What ends up happening over time is that this poly A tail will slowly get whittled away. It'll lose the poly A binding proteins. And you can think of this as a timer where like over time, if this mRNA is not used for translation to make a protein, we'll lose the poly A tail, we'll lose the cap because of that decapping protein, and exoribonuclease will degrade the entire thing. So there's a bit of a time frame for which this mRNA needs to be used, otherwise it'll be degraded. And you can think that if this is a key protein, perhaps like actin in our cytoskeleton or tubulin, if this is a key protein or other proteins that are needed during, say, an immune response, and this mRNA is expressed, it's going to be used if it's important. If not, then it's going to be degraded. So the last thing we're going to talk about is, I'll stop there. Are there any questions for the modifications that we talked about up to this point? Yes.
So when we end this stage, what happens to the end? How is it like broken down?
It's broken down into individual nucleotides, and so those individual nucleotides can be reused for replication and transcription again. Yeah. Yes.
And I know this is a quiz question, but could that still shake on the whole thing with exonucleases? What can you just like refresh us on like what those actually do as part of like the direction and like how those come into play with transcription?
Yeah. So with DNA polymerase, there's this like proofreading function where there's exonuclease activity in a specific direction during replication that would remove non-complementary nucleotides. So like if during replication, there's the wrong nucleotide added on, there'd be like a disruption in the structure of the DNA. And that disruption would serve as like a physical cue for DNA polymerase to stop elongating in the 5' to 3', but rather be an exonuclease in the 3' to 5', but back that way. In this case here for mRNA, the exoribonucleases, they're just looking for a free end to hop on and just completely start degrading. So like that specific directionality of exonuclease activity was specific with DNA polymerase. With exoribonucleases, their sole function in life is just to destroy nucleotide strands. Specific for DNA polymerase. Yes, because DNA polymerase has specific functions in specific directions. And exoribonuclease does not have directionality like DNA polymerase for its exonuclease activity. Like it's not only going to be in the 3' to 5' direction where it's only going to find one end, it's only going to promote degradation in one end. If there's a free end available, it's just going to start degrading the mRNA. I think I saw Justin's hand up, and then I'll go to you.
So the decapping protein first goes on and it takes off with the
No, so there's I'll do the animation again, narrate it. So what ends up happening is that the poly A tail will slowly get lost. And what ends up happening is the poly A binding proteins are lost. And once that poly A tail is lost to a certain degree, the decapping protein will kick off the 7-methylguanine. And then once that's gone, that'll enable the exoribonuclease to begin degrading the entire mRNA strand. So again, it's an exoribonuclease, so it's going to degrade it at the ends. So it's looking for a free end. The poly A binding proteins here and the cap help guard and protect it from degradation. It protects it from having a free exposed end. But it's on a timer. That protection lasts for only so long. And so if this mRNA is not used for translation, it will eventually get degraded.
So can we take the 3' and the slot or
The 3' end is where the poly A tail is. And so that whittling away of the poly A tail is on the 3' end.
got it. And then that's the 5' is the 8.
Yes. Yeah.
So DCP is always found as God. It just doesn't activate until the tail is lost.
Yes. Yeah. All right. Tommy, I'll go to one last question.
So, does DCP associate like once it's left from the nucleus, or is it part of the maturing process, or like after it has its path and then poly A tail DCP protein associates and possibly some folding?
Yeah. So I think that's right. That the decapping protein is present out in the cytoplasm. Because within the nucleus, that's typically where immature pre-mRNA is present. And so if it was present within the nucleus, then a lot of mRNA would be degraded immediately. So the decapping protein, I think, is typically present within the cytoplasm. All right, we're towards the end here. So you all discussed while I was absent about what exons and introns are. Just to quickly restate, exons are typically the coding regions of our DNA, and introns are these regions in between exons that are non-coding. So what ends up happening is that during transcription, both introns and exons are transcribed. But what ends up needing to happen is that to go from immature to mature mRNA, these introns need to be removed or spliced out so that when you go and translate that mature mRNA, you're going to have the important exon coding regions that'll encode for this protein. And typically, certain regions of exons or combinations of exons will usually encode for certain parts of a protein. And so whenever we talk about specific units or subunits of a protein, they typically will encode for one or a few exons that are adjacent to each other. So there might be one exon that encodes for the catalytic domain of an enzyme, and then exon 2 and 3 might encode for the regulatory subunit or the allosteric subunit of a gene. And so the key thing to note here is that the introns during splicing are removed so that we're left with only exons, where the exons are usually what encode for these different domains. And you can see here that's color coded to match the exons. But these exons usually encode for different domains of a protein. So typically what ends up happening is that when we have our primary transcript or our pre-mRNA with our introns, what ends up happening is that they're removed and excised so that the exons are brought together. And typically bioinformatically, each mRNA transcript for a specific gene that's been that's been defined bioinformatically, and it's going to every nucleotide has been we've sequenced our entire DNA, and so we know the transcripts that'll be made from a gene. And so each nucleotide is numbered, but we don't number the introns usually. And so when you go from the pre-mRNA to the mature mRNA, you'll see that it goes from 1 to 146, and with the spliced version, it also goes from 1 to 146 because again, we don't count the nucleotides within the introns here. Now, how does the splicing occur? So there are these specific splice sites that specify the beginning and end of an intron, and it's usually these two base sequences, where on the 5' end of an intron, there's a GU base pair, or not base pair, forgive me. There's a GU base sequence, and on the 3' end, there's an AG. And those are usually, those are conserved across all introns. So those are the splice sites, the signals at which splicing occurs for an intron to be removed. And it's also the target for the formation of what's called the spliceosome, this combination of proteins that come together to splice out those introns. And they use these, what are called SNRPs, these small nuclear ribonucleic particles. And what that looks like here, and this will be our last slide for the day, is that we have a gene where there's an exon to the left and an exon to the right, and we have an intron here. On the 5' end of this intron, we have our GU 2 base sequence and our AG on the 3' end. So this is a signal for an intron that needs to be spliced out or excised. So what ends up happening is that you don't need to memorize what protein binds where. We're going to generally refer to this as the spliceosome, but for full description, I'll say the specific proteins here. So the U1 SNRP will bind to the GU base sequence, and U2 will bind at a specific site that's called the branch point sequence. That'll be important at the end of this process. What ends up happening is that there's going to be, once those two form, there's going to be U4, U5, U6 also joining in, and they're going to start pinching off all those SNRPs together. Where's my mouse? All of these SNRPs together are what we call the spliceosome. So they're going to start pinching off this intron, and what ends up happening is that we're first going to have cleavage here at the 5' end of the intron that's going to come over and bind to that branch point sequence. One thing I want to clarify here is that the image shows like a lot more intron upstream of, or a lot more nucleotides upstream of that GU. That's incorrect. All of this would not be intron. Like the GU are like the first two nucleotides in that intron. So you can like ignore this and assume like that's all still exon. But what ends up happening is that the GU is cleaved and bound over to that branch point sequence where that A is. And then there's going to be cleavage at the 3' end where that AG is, enabling the ligation of the two exons. And here we just have our excised intron that formed what we call a lariat structure, this looping, once the GU is brought over to the A, that branch point sequence. And so here we have a region of the mRNA that the spliced out intron, or the spliced out region of the mRNA that encoded for the intron. There's no 5' cap, there's no poly A tail, so this will be degraded immediately. And so what we have here now is how these different exons are fused together to create a mature mRNA. What's also important to note is that, you know, this is how splicing occurs, how we remove these introns. There's also splicing variants or alternative splicing events where perhaps the AG on an intron all the way over here, like after exon 3 is recognized, so then accidentally, perhaps that's one way in which the version of this protein, or version of this mRNA, forgive me, loses exon 2 and 3. And so the way in which this splicing events occur, this is how splicing occurs. How it's regulated is wildly poorly understood. And so it's a huge area of research right now. But how these introns are spliced out, or how the GU perhaps over here might branch over to another intron way downstream can excise exon 2, maybe exon 3, where you get a different splice variant of the same protein or the same gene here. With that, that is going to end our time for today, but I'll be up here for any more questions. Thank you.